Hey guys, what's up? Mr. Brotherton here, and we're going to be talking about prokaryotes. Okay, this is going to be our first part of our cell unit, so this is going to be 2.1. Okay, so back when I learned all this stuff, they basically just threw you in the deep end with a whole bunch of organelles and no context, and I don't know how anyone made it through biology and still thought it was interesting after that. So my goal here is to give you a little bit more of the evolutionary context for the differences between prokaryotes and eukaryotes. All right, so back in the day, we classified organisms into 5 basic kingdoms, but due to the advances in microbiology and genetics, we have actually reclassified the whole entirety of life into 3 larger domains. And those domains reflect a better understanding of what's happening at the molecular level, specifically with the structures of ribosomes and other DNA parts that all life shares in common. So there's about 500 genes that never really change that all life needs, so we can compare those 500 sequences. All right, so the new classification that Carl Woese came up with in 1977 and took about 20 years before it was accepted looks like this. So we can split life into both prokaryotes and eukaryotes. So prokaryote means before a nucleus, okay, so pro means before, and eukaryote means true or genuine nucleus. All right, so the types of prokaryotes are bacteria, and what Carl Woese defined as Archaea, meaning ancient ones. And then we can define the eukaryotes. We can divide those into a couple different kingdoms. So that's going to be your protists, your plants, your fungi, and your animals, all right? So if you look here, this is kind of the old 5-kingdom classification that I have here. And then so now we break the Archaea and the bacteria apart. They're both still prokaryotes. And then we can kind of break our eukaryotes into these 4 separate kingdoms now. So here's kind of an overview of what that looks like. So you can see here, here's your prokaryote, here's your prokaryotes, and then here are all the different kingdoms of eukaryotes. So sometimes you'll see really old texts that call the kingdom of bacteria the eubacteria, and sometimes you'll still see Archaea called the Archaea bacteria, and this is leftover from the 70s, and it's not really used too much now. So your Archaea are going to be your extremophiles, usually living in geysers and stuff like that. And your protists you probably don't know that much about, and we'll come back to these, but just as kind of a preview, these are eukaryotes, but they're you probably think of them more as bacteria because these are things that generally can harm you, like brain-eating amoebas or parasites, so plasmodia that causes malaria, or giardia. All right. So one of the things that's really cool about life on Earth is we have all this beautiful, amazing, complex diversity, but also there are these things that unify all of life, and those come down to a few structural things. So all life is made of cells. We talked about that at the beginning of class. And so with that, all cells are going to have a plasma membrane. All life is going to have some form of cytoplasm that's filled with this jelly fluid stuff called cytosol. All life are going to have ribosomes that are used for protein synthesis, and all life are actually going to have DNA. One of the interesting things I want to show you here in this phylogenetic tree is that even though bacteria and Archaea are both prokaryotes, meaning they don't have a nucleus, the Archaea are actually more closely related to the eukaryotes. So the Archaea, even though they may look like a bacteria to you and I, and we probably think of them that way, they're actually more closely related to us than they are to the bacteria. All right, so where are prokaryotes? Well, we find these things everywhere. Okay, we tend to find the Archaea in really extreme environments, and you can find bacteria living almost anywhere. Okay, they digest. They can be in your digestive system. They can be living on dead things. They're going to be living on plants and animals. They're going to be living in plants and animals. They're going to be in the soil. All right, so the prokaryotes are a very diverse group. So here's some like super saline lakes, some geysers and stuff like that. Here's my cat. She's being obnoxious. All right. So they always find a way to make it work. Here's just a whole bunch of differences in bacteria, what a bunch of different bacteria look like, and they can be classified that way. All right, so what's going on with prokaryotes? Generally, your prokaryotes are going to be unicellular, unlike eukaryotic cells, which can become multicellular organisms like you and me. Prokaryotes are also really, really small. All right, they are going to be about 1/10 the size of a eukaryotic cell, so only about 1 micron across. Their internal structure, this is really the big difference. They have no internal compartments. They're not going to have any membrane-bound organelles. They're only going to have ribosomes. That's kind of the only organelle they're going to have. And they're going to have a circular chromosome, which we'll talk about here in a second. But basically, they have naked DNA that's not protected by a nucleus. So it's not going to be, and it's also not going to be wrapped around proteins. And you can kind of see the big size difference here. This prokaryotic cell is really small and this eukaryotic cell is big, and it has all this other complex stuff going on inside of it. All right, so there's a couple of different prokaryotes and what they look like. And then this down here is actually showing you what their DNA usually looks like. So it's usually in these circular things, and that'll be on the next slide as well. All right, so what's going on with the chromosomes inside of eukaryotes versus the circular thing in a prokaryote? Well, generally eukaryotes have a lot more DNA than prokaryotes, up to 1,000 times more, and it causes issues. So in a prokaryote, like E. coli, their circular piece of DNA is about 1,300 micrometers across, and like I said in the previous slide, they're only 1 micrometer across. So that's a serious packaging issue. And how are we going to make that work? So in a prokaryote, they just really condense that down. Just for an analogy, that's the equivalent of taking 60 feet of thread and trying to fit it into a thimble. Okay, so that's a hard thing to do. And now imagine in a eukaryote, you're compounding that problem by a lot, because if you have 1,000 times the DNA that's in a prokaryote, that's a serious packaging and logistical nightmare. And eukaryotes have solved this by wrapping their DNA around these histones, and that's how you get these chromosomes. So these things aren't going to be packed around proteins. Our DNA is going to be packed around proteins to help it stay condensed and organized. All right. One of the things that separates Archaea from bacteria is that they actually do have some histone-like proteins. They still have that circular DNA, but that shows us that they're actually more closely related to eukaryotes than they are to bacteria. All right, this is really cool. The variations in the cell interior of our bacteria can actually show us a little bit about the evolutionary history of eukaryotes as well. So here is a comparison of an aerobic bacterium and a cyanobacterium, and then it's also showing you the organelles that are inside humans, or animals and plants and all eukaryotes, which are the mitochondria and chloroplast. So what's really cool is that generally there aren't internal membranes in bacteria, but in a cyanobacteria they have they have a lot of these folded over membranes, and that's where photosynthesis is taking place. And guess what? That looks almost identical to a chloroplast, which shows us that at some point in time, early eukaryotes actually engulfed one of these cyanobacterium and formed this mutualistic relationship, and that's known as the endosymbiosis theory. Same thing happening with the mitochondria that you will find in every single eukaryote, including plants and animals. Same thing, this aerobic bacterium also has a very similar structure to a mitochondria, and when we actually examine the DNA of mitochondria, its closest relative is actually a parasitic bacterium known as Rickettsia. So it looks like at some point in time this thing tried to invade a different cell, and they ended up forming this mutualistic relationship, and that's what led to us. And the same exact thing with cyanobacteria and chloroplasts. If you examine chloroplast DNA, its closest relative is actually cyanobacteria, not the nuclear DNA of the plant. Same thing with the mitochondria, not the nuclear DNA of the plant or animal that it's in. All right, so what's going on with the cell structure of prokaryotes? They pretty much come in two different types. at least when we're talking about bacteria. So you can either have what's known as gram-positive bacteria or gram-negative bacteria. You've probably heard that before at the doctor maybe. And that comes down to a staining. So if it's gram positive, it turns purple. If it's gram negative when it's stained, it turns red. What is that telling us? Well, it's actually telling us about the structure of the cell wall there of our bacteria. So a gram positive bacteria is actually less resistant. That's why it shows up purple. And a gram negative bacteria is going to be significantly more resistant. When I'm talking about resistance, what am I talking about? Well, I'm talking about environmental stresses, but I'm also talking about antibiotics. So a gram-positive bacteria is much easier to kill, whereas a gram-negative bacteria is much harder to kill, as you can see here, because it has this extra outer membrane of these kind of fats and sugars. One thing that's pretty cool is that once again, Archaea are a little bit different, and so they actually don't have these peptidoglycans, and that's another reason we know that they are different from bacteria. All right, so if you think cellular respiration and photosynthesis are complicated, you haven't checked out prokaryotic metabolism. These things are crazy. They are very, very good at finding ways to live that we are not familiar with at all. Okay, generally they can be photoautotrophs, so that's going to be like your photosynthetic bacteria, which are your cyanobacteria. And then you also have what's known as holobacteria. Technically, they are an archaea. They do a weird form of photosynthesis that doesn't actually use oxygen, which might be the oldest form of photosynthesis on planet Earth. Prokaryotes can also oxidize inorganic compounds. So these things can live on nitrogen, sulfur, hydrogen. They can actually eat iron. I mean, they're crazy. And then other ones can also eat the same way you and I eat. So they can be heterotrophs as well. Plant and animal matter they'll eat. And these are going to be your decomposers and the things that get you sick, so pathogens. All right, so genetic variation in bacteria. Bacteria can reproduce every 20 minutes through binary fission. Their error rate is relatively high, and so they can mutate pretty quickly, and that's why we end up with things like antibacterial resistant bacteria, because they can mutate and change pretty quickly, and they can. What's really cool about bacteria is they can actually kind of swap genes, and they have these small little extra supplemental parts of DNA known as plasmids, and those are really pretty cool. So they can like store information about things that have attacked them in these plasmids. And also they can do another thing known as horizontal gene transfer, where they can kind of swap genes with other bacteria, and that leads to all kinds of recombination. Okay, so whereas we eukaryotes tend to do a sexual form of reproduction to get genetic variation, bacteria can get genetic variation in other ways. So some bacteria are really bad for you and can hurt you. You probably know about that. These are all the things that they can do. So STDs, TB, Lyme disease, you've probably heard about, tooth decay, okay, fruit rot, wilts, all of those things can be caused by bacteria. At the same time, we need bacteria because they decompose, so they're recycling the nutrients on Earth, so they also fix nitrogen, so we can't actually access nitrogen. Bacteria have to fix that nitrogen in a form that we can use, and then plants and animals and other eukaryotes are able to use it. Also, they can help in digestion. So what's really crazy is that for every cell in your body, there's about 1.5 times as many microbes to every one of your cells. That means that you're really this collection of a lot of forms of life, and you need those to help you digest. And we talked about how cellulose is digested in herbivores by bacteria. All right, so that's all I have for you guys today. Thanks a lot, and stay curious. Bye.